So good evening, everyone, and thank you very much indeed for coming along for this inaugural lecture of Professor John Thornton. I'm David Werring. I'm the head of the Department of Translational Neuroscience and Stroke. So a very good evening to colleagues, friends, family members and other distinguished guests this evening. And it's a real pleasure to introduce this inaugural lecture for Professor John Thornton. I expect you're getting used to being called that now, John, because it was a while ago, and he's been a trusted colleague and friend over many years here at UCL. So most of you will know the format for these inaugural lectures, but just to remind you, first we have a few short remarks from me, about half an hour perhaps, and then John will give his inaugural lecture, which is probably around 50 minutes or something, John, maybe. And then we'll have a vote of thanks, which this evening is going to be delivered by Professor Tarek Yousry, head of the Division of Neuroradiology. And then I'll just give finally a very few short remarks, which will be mainly where to find the drinks at the end of the evening. So we're celebrating tonight the appointment of John Thornton as Professor of Clinical Magnetic Resonance Physics. And his lecture is entitled Magnetic Resonance for Measurement. And on reading up on what John's done and being familiar with some of the work that he's done, this is not going to be just about scientific progress, but what seems to be a personal philosophy that's guided his career, that MRI is more than just making images, but it's about making measurements of tissue properties that are relevant in understanding and treating neurological diseases. So just in brief, John will tell you about the story of how he got here, but he earned a Bachelor of Science degree with honours in physics with electronics from the University of Manchester in 1986. He went on to complete his PhD in instrumentation for MRI at the same institution in 1992. Hope this is all correct so far, John. And then in 2013, he expanded his expertise by getting a postgraduate certificate in leadership and service improvement from the University of Manchester Business School. And he joined the National Hospital in 2003, the Department of Neuroradiology, as its only NHS UCL MRI physicist. And since then, he's built a UCL research team and a group of dedicated NHS healthcare scientists. And I think over the past 2 decades, really, Professor Thornton has become a quiet but powerful force behind some of the most impactful advances in clinical MRI applied to neurological conditions. And he's so modest, he won't tell you this, but his work has not only advanced the study of MRI, but has also enabled a very large number of collaborative projects across different diseases, disciplines, and institutions, both in the UK and globally. Very briefly, just a few areas which John is going to talk about, no doubt. So firstly, measurement as a biomarker, muscle MRI in neuromuscular disease. I think this is perhaps one of John's most influential contributions among many, is developing quantitative muscle MRI as a biomarker for disease progression. He's got a great number, probably over 30 or so senior or co-author publications on this topic. He's really pioneered quantitative MRI to track disease progression in diseases such as inclusion body myositis, ALS, with MRI measures targeting muscle assessment. But John will tell you much more about this. This development has allowed multi-centre clinical trials in conditions which have allowed us to define objective reproducible endpoints, for example, in Charcot-Marie-Tooth disease, with a muscle outcome over 5 US and European sites. And these aren't just technical developments, they're deeply collaborative with neurologists, physicists, neuroradiologists and patient communities across continents. Now, I'm going to get to say the rest very quickly to allow John to get onto his inaugural lecture, but the second area is quantitative neuroradiology, having founded the Quantitative Neuroradiology Initiative, a cross-faculty collaboration in UCL, involving the CMIC, Centre for Medical Imaging Computing, the Neuroradiological Academic Unit and the clinical partners. Thirdly, magnetic resonance safety. So this is a really important contribution to pioneer safe MRI protocols for people with DBS, deep brain stimulation, and other intracranial electrodes, which has allowed life-changing treatments and groundbreaking research into DBS. And then just finally, most importantly, John is a true collaborator, enabler, educator, and mentor. And he's known not just for all this research, but his generosity as a collaborator, supervisor, mentor, and teacher. And he's done rather a lot of postgraduate teaching, actually, including co-establishing the MSc in advanced neuroimaging. So I think, in summary, Professor Thornton's work exemplifies integration of technical innovation, clinical relevance, and patient-centred care. And it's really been instrumental in allowing MRI to be applied to real clinical challenges. That's real translation. And very proud of that as the head of this department. So his inaugural lecture is going to explore his journey of quantitative neuroradiology from concept to clinical impact, at least I think, and hopefully some perhaps embarrassing pictures of you when you were younger, John. I don't know what we're going to see, but very much looking forward to it, and I welcome Professor John Thornton to the podium.
Thank you very much, David, for those words. And luckily, those are the things I'm going to talk about. And thank you, everybody, for coming, and for those that are watching online or on the recordings. So I'm going to talk, as David suggested, a little bit about my journey to here, to Queen Square. I'm going to talk about something about some of the MRI physics that we do and have done in the National Hospital here. And then I'm going to talk about a couple of the main research areas I've been involved in, using magnetic resonance for clinical measurement. So this all started for me when I was about 7 or 8 years old, which is roughly in 1972, giving it all away, when I was a pupil at Elmrose Junior School in Bournemouth in Dorset, where we lived. And we had a really good junior school teacher called Miss Jeans, who in her classroom set up a science table and allowed us to bring in various sciencey things and gave us various sciencey things to work with and explore and kind of start to learn about the ideas of science. And as you can see, at this time, this book, this Ladybird book of magnets, bulbs and batteries was really meaningful for me. I've still got my copy. You can see that I stamped my name in it because it was important to me. And it encourages us to do experiments like this. So times have obviously changed because you wouldn't really be recommending kids to do things like that. But we did, and we survived. And there was this group of boys. So at the time of the times, it was a group of boys, and as we'll see later, fortunately things have changed. But we learned together. We shared things we learned about how to make a bulb light with a pair of scissors and a battery and all these things. And that's really where science started for me. As I moved to the next level of schooling, in my pre-teen years, I continued an interest in science. I actually had that chemistry set, which my grandfather possibly somewhat mischievously gave me for Christmas. It says on it safety tested. I'm not quite sure what that was supposed to mean, but I had a lot of fun with that, setting fire to things and making smells and learning a little bit about chemistry. I was interested in natural history. I did discover that the common newt can indeed climb up the plastic walls of a tank it was in my bedroom and find its way to the carpet of the downstairs living room quite successfully. And I also became very interested in electronics through another Ladybird book, which taught us how to make crystal sets and hang long wire antenna down the garden and pick up radio signals. So this was all, hands-on science at a very young age. So I'm not going to go through every stage in my life in this detail, but so to skip forward, I attended the Slate Grammar School in Bournemouth, which I really think I benefited in educational terms from, and then moved on to A-levels at Farnham College, and then found my way in the end to university in the north, to the University of Manchester Institute of Science and Technology, which you can see here sitting in its post-industrial location, and that really summed up the ethos of the place. It was an institution that came from the Industrial Revolution, and really this course gave us a training in experimental physics and making the skills needed to make measurements, as well as the usual things about pure physics. And this really stood me in good stead for the science I was to do later. So, having completed my undergraduate degree, I had to do something else. And at the time, the options for people interested in physics or people graduating in physics and electronics were the electronics industry or the defence industry, neither of which particularly appealed for me. And I had this idea that I'd like to do something involving medical physics. And in the end, I was offered an EPSRC-funded PhD studentship to work in this method called NMR imaging, which at the time I'd never heard of. So this is a long time ago. MRI really wasn't the thing. And I had no idea about this, and this is one of those forks in the road in life, which turned out to be very fortuitous because it was a good choice. So to briefly, I know most of you in the room are very au fait with what MRI imaging is about and how it works. But just to go right back to basic principles, the nuclei of certain atoms, in particular the nuclei of hydrogen atoms, of which there are a very high concentration in the body, they have a magnetic moment, which means they behave like miniature compass needles, and when they're placed in an external magnetic field, on average, they tend to line up with that field. If we then insert a burst of radio frequency energy and knock the nuclear magnets out of alignment with the field, they undergo this motion called precession. They rotate around the direction of the magnetic field like this, and this is the same sort of principle as a gyroscope precessing in the Earth's gravitational field. It's the same physics. And the really important thing is that the rate at which they precess, their rotational frequency, is directly proportional to the strength of the magnetic field. And this is the key result which enables all the measurements we can make with MRI. If we place a coil of wire close to these rotating magnets, they induce a voltage in this coil of wire by Faraday induction, which with a suitable measuring device, maybe not like this, but that's the idea, we can measure this voltage and the frequency of the signal, the time delay between the peaks in this signal, depends on the rate at which the nuclear magnets are spinning, which is directly proportional to the magnetic field strength. And this is the entire basis of how MRI works in a sort of in a teaching explanation. And when I started my PhD in Manchester, I was very impressed by this paper by one of my supervisors, Ian Mackenzie, who laid out, it's called Nuclear Magnetic Resonance for Medicine, nuclear magnetic resonance for measurement. And he laid out how this phenomenon of nuclear magnetic resonance could be used to measure a whole raft of useful things. And it's kind of lays out the basis of quantitative magnetic resonance at a time in 1986 before MRI was really a thing, so it was quite prescient. Because the precession frequency, the frequency with which the nuclear magnets rotate, is proportional to the magnetic field strength, if we make the magnetic field strength vary with position in space, then a nuclear magnet in this low magnetic field will spin slower than one in the high magnetic field, and therefore this one will emit a frequency which is lower than this one. So if we can perform a frequency analysis of the resulting signal, we can work out where in space those particular spins are. And that's how we form the MRI image at its most basic level. After we've knocked the spins out of alignment so that they are precessing, over time they lose energy, they exchange energy between themselves and they lose energy to their environment. So they tend to revert back to being in alignment and not precessing. And the rate at which this happens is a function of the amount of energy exchange between the nuclear magnets and their environment. And this is influenced in human tissue by disease conditions. So if we can measure these so-called relaxation rates, we get information about what's going on in the tissue. We can also, by more sophisticated methods, we can make the signal sensitive to flow of the material, so to blood flow in the body, and to self-diffusion into the speed, the average speed of the random Brownian motion of water molecules. And we can also perform chemical spectroscopy measurements, because the hydrogen atoms in different positions on molecules experience slightly different magnetic fields influenced by the electron clouds of the molecules. We can separate them in the signal, and we can do a chemical analysis of what's in the sample. non-invasively. So all of these things were possible then, and I'm going to show some examples of all of these as we move forward. So back to the PhD. So the title in the end of my thesis was low-cost, high-res instrumentation for low-cost, high-resolution NMR imaging system, and low-cost wasn't joking. So the magnet we used was this thing in a picture on the left-hand side, which was a water-cooled electromagnet, 0.8 Tesla. And the idea was to be able to place a sample in between the pole pieces of that magnet, the gap in the middle, and be able to scan it, to image it. And this was all home-built electronics in the Department of Instrumentation and Analytical Science in UMIST in Manchester. And my part of this was firstly to characterize the magnet. So I spent long hours moving a probe around between the pole pieces to plot how the field varied in space and generate with the crude graphic systems that were available at the time, making contour plots like this of the magnetic field as it moved around, and then to make the gradient system. So I started off just before my PhD, looking fairly fresh-faced, and all was good. And then I went on to work on the gradient system design. So I designed the digital controller for the gradient system using a BBC microcomputer, which was current at the time. I designed some the gradient amplifiers, and I designed the gradient coils. So in this system, for the gradient coils, they had to fit against the pole faces of the magnet to generate the imaging magnetic fields. So we decided that we were going to do this by making high-density printed circuit boards to form the conductors. And I did some finite element modeling to optimize the arrangement of the conductors. And you can see some of these structures within the magnet there are experimental gradient systems. So it was all a lot of fun, but you can see that towards the end of my PhD, it may have taken its toll. Or maybe there were some distractions in Manchester around that time, I don't know, but this is what the PhD can do to you. And at the end of the best part of 4 years, we finally managed to do at least a vaguely credible imaging experiment. So the test object that was put in the pole pieces of this magnet were 2 capillary tubes here with some water in the bottom, so essentially 2 blobs of water. And I managed to get the scanner to come up with something that looked at least plausibly like 2 blobs. And that was the result. And I'd run out of time, and we'd run out of money. There was no funding. It was, And as you can see, I think I've really had enough by this point in the project, and it was time to get a job. So I collected enough data to be able to complete my thesis. And I answered an advert in the New Scientist, which was how jobs were advertised in these pre-internet days, for a postdoctoral position at University College London. And again, I hadn't heard of any of this. It was just an advert, and I applied for it, and in the end, I got the position. So this was in 1990. So this is the start of my association with University College London, which is 35 years ago, which is something. And I joined the Department of Medical Physics and Bioengineering in David Delpy's group, and joined there was a major push in collaboration with the neonatologists who had their departments over the road, to join a group that was investigating using different techniques, magnetic resonance, infrared spectroscopy and other techniques, to assess, in newborn infants, to assess the effects of hypoxic ischemic encephalopathy, which was a culture shock for me in some ways, or the first culture shock in this story. And when I arrived there, the group was quite long established, and Ernest Cady was leading on the spectroscopy, and what they were routinely doing were taking human newborn infants and putting them in a Brucker spectrometer. So this is not the actual one we use. This is a 70 Tesla scanner we had in the lab next door, but the scanner we used, the magnet we used for the babies looked essentially like this. The babies were brought down from the neonatal unit in a transport incubator like this, in a Perspex tube, and pushed into the bore of this experimental magnet, and the experiments proceeded. And looking back on that, this is quite a thing. This wouldn't happen now. But it was hugely influential work led by Osmond Reynolds, who at the time was the father of neonatology in the UK, and led to lots of good things. But this was already established, and my role when I got there was to work up imaging, because they hadn't done any imaging on the system they had then at the time. So that was my role. And there are lots of colleagues from that time that I'm still in touch with. In fact, this is when I first met Marjana, who's now in a team here, because Marjana, you were doing your PhD with Ernest Cady at the time in this lab. And other names that will be familiar to some of you were also there. So this, for me, was an exposure to a very clinically applied application of MR physics, much more so than you would normally get exposure to. And one of the pieces of work that is closest to my heart that I did was to work up T2 relaxometry in the newborn brain. So you can see the image quality is improved. The images were quite good on that system. And I programmed the MRI pulse sequence to collect a multi-echo 4-echo sequence with crusher gradients and optimized slice profiles to get very accurate readouts of the decay rates of the signal, from which we could estimate this parameter called T2, which is essentially sensitive to how watery the brain tissue was. And the reason we thought this would be interesting is because we know in the newborn brain that the newborn brain is obviously very different to the adult brain. There's a process of maturation which essentially involves at a very crude level, the brain becoming the brain tissue becoming less watery as the nerve cells proliferate and migrate and become myelinated. So this was really a measurement of this, and we were really quite surprised to find that from this yellow region of interest here, that the T2 of the brain tissue was decreasing by about 2 milliseconds per day, which is a measurable amount, and obviously this would have impact for clinical imaging of neonates, for optimizing the sequences, and also for when we move this experiment to human infants. So, not to human infants, when we move from babies that parents, very healthy babies that parents very generously consented to allow us to do these measurements on, to scanning babies who were term infants that had a challenging birth, and therefore were at risk of brain damage from hypoxic ischemic encephalopathy. And in this study, we scanned a number of these infants and measured this T2 measure in their brain, and the values from the healthy infants are down here. They're quite low, and the values from the brain-injured infants are quite high, are up here. And you can see there's not separation, but clearly this value is giving an indication of outcome, and this is the 12-month follow-up outcome. So this is the neurodevelopmental outcome at 12 months following the original scan at 1 year old. And you can see there was a relationship, but actually, as you might expect, magnetic resonance spectroscopy was a stronger indicator of what was going to happen over 1 year, because it directly measures the metabolism. However, this is a good example of the potential for quantitative MR to measure something that can be used to inform patient management, in this case to help with counselling the parents and to help the clinicians potentially make difficult decisions. And this really stuck with me as something that was worth pursuing and potentially very useful, but also I was very uneasy about how this could be translated into something that could be used more universally, that could be taken up in other centres and used by other people. So I'll come back to that. Also during this time, my first collaboration with folk from here at the Institute of Neurology was with the MS group, with John Mottershed, who was one of the clinical fellows at the time, who brought across ex vivo post-mortem human samples of human spinal cord, which we scanned in our 7T system there. My role was to program all the pulse sequences and do all the data analysis, all the pixel bars fitting for the MR parameters. And the purpose of this was Firstly, to show that we could measure these quantities in ex vivo spinal cord, but also then to relate the MR findings to histopathological findings so that we could see what the underlying pathological substrates of the MR changes were. And this was quite successful. We showed that magnetization transfer related quite well with myelin content, and that T1, for instance, there was a good correlation with axonal density. So this turned out to be quite a well-cited paper because there weren't many papers available at the time where this kind of corroboration work could have been done. So when my time in medical physics came to an end, I had a brief and very enjoyable stay just around the corner at the UCL Institute of Child Health in David Gadian's lab. So I should have mentioned before, Roger Ordich was mentioned on the previous slide. Roger was really something of a mentor to me, and he helped make the connection for me to move to David Gadian's lab, where I wasn't there for very long, but it was a really good time, and the team there were great. And my role here was to set up time-of-flight angiography on an animal scanner. So I programmed the pulse sequences with the gradient moment nulling and worked out how to do these projections. So these are views of the arteries in the neck of rats and in the brain. And this was a preclinical model of occlusive cerebral arterial disease. So the arteries, some of the arteries had been closed off, and the animals were scanned 6 months later, and fairly remarkably, the blood perfusion to the brain had recovered. And it recovered in large part because there was an adaptation of the arteries feeding the blood supply to the brain during that time. And we were able to show that. And we weren't measuring blood flow, but we were measuring blood vessel size. And this was an interesting precursor to where I would be going next. And where I would be going next is here. So I come to the end of my time as a postdoc. It happens to quite a few people. I've been really well supported by senior people who looked after me really well and found grant funding to keep me going. But that time had really come to an end, and I knew that I would have to leave London, leave UCL, or find another alternative. And Roger Ordich, again, said to me he knew this chap called Tarek Yousry who was trying to set something up and might have an opening available, and I should ask him about it. So I did get in touch. And eventually Susan wrote back, Susan Wakely, and made the connection. I came and looked around the department when Helen Gunson showed me around because she was the only senior radiographer available. And I ended up taking the position at Queen Square. So at that time, there was just one MRI scanner here in the hospital, and there was just one clinical scientist, one MR physicist in the hospital, which was me. Now we have 5 state-of-the-art Siemens MRI scanners, and there are 6 clinical scientists working in MR physics and scientific computing. So things have expanded. And with all our fingers crossed, in early 2027, we will move into we will also absorb the new imaging facilities in the new Gray's Inn Road Neuroscience Centre, where there will be a further 5 MRI scanners, and we will obviously have to expand our staff to accommodate that. So there's been quite a change over the years, and the change will continue to happen. In case you don't know what a clinical scientist is, or what an NHS clinical scientist is, we are PhD level scientists who've done training in medical physics. We're accredited by the Health and Care Professionals Council in the same way as radiographers and therapists. And our role in the hospital next door is to provide expert scientific and engineering support to the MRI service. What that means in practice is our mission is to benefit patients, the patients coming through our service now, by undertaking all of these activities, advanced MRI and DTI, MRI support, MR safety support, making sure that we optimize the use of our scanners to increase patient throughput as much as possible, and the other activities you see here, including capital projects and MRI equipment procurement, which also supports the research infrastructure. So those are the things we do to benefit patients passing through the service now. And we also work to benefit patients in the future by supporting, enabling and leading MRI research. You can see us there a few weeks ago, just after we've been for lunch, which is probably why everyone's smiling. Our current members, Matt, Annie, Lara, James and Marjana. Jolie is not in the picture because we've just appointed her and she will be joining us in October. Our former members, one of whom is here today, left us to take more senior positions, Mark White and Stephen Westling in our trust, and Enrico De Vita leads the clinical MR physics team in the Great Ormond Street Hospital next door. And also our UCL research physicists are also in the picture here, Amy and Nick. And I'm really proud to lead this team. They do this work very well, very professionally, and are in some ways the epitome of a good team. And thank you very much to all of you. So, one of the areas when I was trying to work out how to make myself useful as the only physicist in the department before Lara joined in the first year was to look at MR safety. And Marwan Harries, who was a pioneer of DBS functional neurosurgery, had already established protocols with the radiographers for scanning patients with DBS electrodes. But we need to do some more work on it. So deep brain stimulation is a life-changing therapy for people with Parkinson's dystonia and other conditions, which involve inserting electrodes very deep into the brain. The problem with this is that this then creates a significant safety hazard if these patients need to undergo MRI, because the radiofrequency fields of the scanner could induce voltages and cause heating at the end of the electrodes. So we started a series of studies over the years to work on this. The first one was with David Carmichael and Louis, who's here. And there were a series of papers and studies that followed this. And the idea really was to take a DBS system, as would be implanted in a patient, and implant it in a big tank full of gel, which simulates the human tissue, scan it, and use fibre optic temperature sensors to see what the temperature rise would be. And in this particular study, when we did this, we measured a 1.4 degrees C temperature rise at the electrode tip when we ran a standard diagnostic MRI scan. So temperature rise is not out of control, but it's high enough to be worried about. So this enabled us to design MRI protocols to reduce this heating and offer safe MR to patients with DBS systems in situ. This had two benefits. The first obvious benefit is to our service to improve the scanning for patients coming for their DBS therapy, but also to enable research. So there were also then, following on from this, we improved our safety assessments, and this enabled collaborators, in this case studies led by Tom Fultney and his student Josh Gahan, where it was now possible to scan patients with DBS systems in situ and perform functional magnetic fMRI studies to look at regional activation in the on and off stimulation condition, which started to give the neurologists involved an understanding of how DBS might actually be working and the effects it was having on the networks in the brain. So this is an example of MR physics being used to enable the research of colleagues, high-impact research. And I should mention also Louis' main interest is in EEG MRI, and we also did a series of studies on this, which also benefited the patient service for patients coming for scans who have EEG electrodes in situ. But that's nothing to do with magnetic resonance measurements. So if we get back to the topic, one of the areas in the earliest phases of my time in neuroradiology was collaborations with the prion unit, where we used quantitative MR methods to measure changes that were going on in the brains of patients with prion disease. At the time, there were no useful biomarkers for prion disease. I think obviously things have moved on since then. But we set out to do this, and this was with a couple of PhD students that I co-supervised, Harpreet Hiari, many of you will know, who's now a consultant in our department and is in our research department as well. And my role here was to set up the acquisition protocols and do the computational image analysis, and they did all the rest of the work. But we showed, for instance, that measuring this quantity called magnetization transfer, which is quite abstract. It's a measure of the the communication of magnetic energy between free water that can move freely and water which is attached to membranes or in the hydration shells of macromolecules. And as the brain's tissue starts to disintegrate, this drops because there are less macromolecules there. And we showed in Durry's PhD thesis, she showed that there was, for instance, in the top graph, a decline in the whole brain MTR histogram metric correlated very well with a decline in clinician's dementia ratio, the clinical assessment. So there was something going on. And then in post-mortem studies, we showed that this decline in MTR in post-mortem tissue correlated with histopathological measures of spongiosis. So this indicated that we were measuring something that had biological relevance. There were a series of papers that came out through the following years where we tried different techniques, mostly led by Harpreet and some by Enrico, who was doing the analysis with us. And the final one was in 2020, a paper that Harpreet led on, where we were looking at self-diffusion measures measured by magnetic resonance, and showed that we could correlate these MR measurements of, in this case, radial diffusivity, which is one of the measures of anisotropic diffusion, and in quite a heterogeneous group of patients with prion disease, which included both inherited prion disease and sporadic CJD, that this has promise as a biomarker. And we did some calculations to show what this might mean in terms of the numbers of volunteers required for trials if this was used as an endpoint in a clinical trial. And it held up both at the region of interest and at a voxel level analysis. So this was a really encouraging example of magnetic resonance measurements that were measuring something possibly important. But these were measurements of fairly abstract quantities, which are not always, it's not immediately obvious always how to interpret what they mean in terms of the underlying pathology. So the next area I'd like to talk about, which has become one of my main areas of focus over the years, is performing magnetic resonance measurements in neuromuscular diseases. And the very first study we did of quantitative MR in muscles we started in 2010 with Mary, where we studied patients who had neuropathies, so damage to their peripheral nerves, which is called denervation, which causes the muscles to waste. I'd like to say we decided to use magnetization transfer ratio because we had a strong hypothesis that there'd be a difference in the magnetization transfer. Actually, no. Magnetization transfer was the only quantitative MR technique we had on our GE 1.5T system because it had been implemented in the pulse sequence by Gareth Barker. And it's the same sequence that we used in the previous studies of prion disease. So really it was just a let's see what happens kind of study. And as you can see, there was a difference in the magnetization transfer ratio readout between healthy control subjects and patients with the inherited neuropathy CMT1A. And also there were some interesting patterns of change. But what we noted at the end of the study was that fat infiltration complicates interpretation because there's a lot of fat in the muscles of these patients, and fat generally doesn't display magnetization transfer. So this was clearly a confound. And we did quite a lot of interesting work with Xavier Golay, improving the magnetization transfer measurements and doing proper quantitative magnetization transfer. We kind of abandoned this line of research and instead went on to actually measuring the fat content rather than it being a confound, using it as the main readout. So I was aware at the time of this method called Dixon fat water imaging, and I asked Chris Sinclair, who was our postdoc at the time, to implement it, and he did a really good job of implementing it. We were still basically using the method he set up. So this is a method of 3-point Dixon fat water measurement. And this exploits the fact that the processing nuclear magnets in different chemical locations produce signals at different frequencies. So the water signal is at one frequency and the fat signals at another frequency. Because of this chemical shift effect. And if we collect images at different points in time, then the water signal and the fat signal, sometimes the water's down and the fat's up, and sometimes it's the other way around. And if we collect these three images, we can exploit this and perform a mathematical inversion of this to extract an image which just represents the water concentration at each location, and an image that just represents the fat content in each location. And we can use this to measure something that we call the fat fraction, which is the proportion of fat to water at each location in the tissue. And then we can perform a segmentation and get a measurement from each of the muscles, for instance, of how much fat there is in those locations. And this is a very powerful thing. It turns out that this is almost perfect for a magnetic resonance biomarker because it's something that's easy to understand. It's something that's obviously clinically important. It's something that in diseases shows a large effect size. The changes are quite considerable, often in disease. And it's something that we're measuring directly. We're measuring this based on it's a chemical measurement performed by magnetic resonance. We're not inferring it from some complicated abstract model. So, and this is one of the reasons that this has been, as well as the fact that it's just practically worked out, that this is so successful. And now this method is a key method in neuromuscular diseases research. And these are some results from a landmark paper that Jasper Morrow led on, that we published in 2016. Where you can see here in a cohort of patients with the neuropathy CMT1A that at the baseline that their muscles are more fatty, you can see it, and we could measure it and show that in controls the muscle fat level is very low, and in the patient group it was significantly higher. And then looking at change with time over 12 months, the patients really didn't change very much at all. The controls, the healthy individuals, didn't change very much at all, whereas in the patients there was a measurable change of fat fraction. And in this particular example here, you can see over 12 months that you can't, eyeballing it, it's not easy to see, but performing our quantitative measurement, we could show that the amount of the fat in the muscle had changed from 26.8% to 28.8% in this individual. So it's a very powerful technique. And this year, Jasper and Matt, under Mary's direction, published data from the same group of patients who were studied at a 4-year time point. And the data showed that the changes we measured by MR at 12 months correlate with the changes in clinical performance that are measured at 4 years. So there's a direct relation, and at 12 months, the change in the functional measures is really too small to be useful. So this is showing us that our short time scale MRI measurements are predicting the long-term clinical changes which are important to patients, which really reinforces the value of this method. To help us along the way, AI happened. Deep learning came as a tool that we were able to use, and I attended a BRC, a meeting of the healthcare engineering and imaging theme, and presented our work on neuromuscular imaging. And this led to a collaboration with Baris Kamba, who's a computer scientist who was working with neural networks, and he said, I can help you with this. So we already had a large archive of images that we'd done this kind of a manual analysis on that we'd used for the papers we'd published in different diseases. And we were able to use this, Baris was able to use this to train a neural network to do this process for us automatically. So now we no longer have to spend a huge amount of time manually drawing around these regions to extract this data. It can be done by the computer, and it's actually quite reliable, and it's something that we're continuing to improve in our current research. So this is, and we're using it now in our patient studies as a tool to accelerate the process. So because the accumulation of fat in muscles is a common pathway for many different neuromuscular diseases, this has been really useful. I've had the privilege of collaborating in a large number of studies, of different diseases with different collaborators where we've used this method and shown that it's a potential sensitive outcome measure. And the focus is shifting in some ways to actually being able to deploy these imaging biomarkers in multi-center trials of new therapies. So to get to a state of treatment trial readiness as new treatments emerge. And just this week, it was officially announced that Mary, under the leash of Mary Riley, we have a grant of £1 million from the Muscular Dystrophy UK to set up set up some trial readiness across the UK, including imaging to run at several different centres across the UK. And we're going to improve our protocols and our pipeline for doing this as part of the process. And this was announced on a couple of days ago, and much to my surprise, here we all were on a photograph of the website with Mary in the middle, and next to her is Amy McDowell, who's going to be the lead MR physicist on this project. So I never quite left the idea of these T2 measurements, which I did in the baby's brains, came back to that. And it turns out that these measurements are much more difficult to perform in fatty muscle, but we've spent some time and energy trying to do this. And we see, for instance, in denervated muscle, in neuropathies, that we see a pretty much universal elevation of T2. The control subjects are blue. They have a low T2 around 30 milliseconds, and the patients are more yellow. They have higher T2s fairly universally. We see changes over 12-month intervals, which are indicated by those blue lines in that plot there. But we think these are probably subclinical changes. But we're still looking into that. But in other conditions, for instance, inclusion body myositis, in muscles that are much more severely disrupted, we actually see a T2 decrease, which complicates the interpretation. So in these examples from this publication, from a study led for UCL by Pedro Mercado and sponsored by Pfizer, that in the early stage disease, the T2 is a higher level than we'd expect in volunteers, and in advanced disease, the T2 is lower. So there's something complicated going on in terms of replacement of the different compartments. And the clinical value of this is still not yet clear, but we'll keep trying. We'll keep trying to improve our measurements. So this is just a slide to acknowledge most, I hope, or many of the collaborators that have contributed to these studies that I've been privileged to be a collaborator in. I've probably forgotten some people, but these are, it's been over 15 years now. It's been a major endeavour. But it's been very satisfying to actually get very close to achieving magnetic resonance measurements in human patients that can be used to test new treatments. So this is something we think is a success. So we can do these measurements usefully for certain clinical applications, and we can show that in groups of patients, we can measure differences between groups of patients and groups of healthy people. Works at the group level. But sometime around 10 years ago, Tarek and I came from different professional viewpoints, came to the same question of why can't we use these types of methods in neuroradiology practice? Why can't we use them in the hospital to evaluate patients coming for MRI scans and incorporate the information into their reports? Because they're so useful in research, and we've all worked on that. We've all published papers saying these are potential clinical biomarkers. So there are many things we can measure with MR, but they're rarely used in neuroradiology practice, I should have said. There's a great counterexample in measurements of the liver in general radiology, but in neuroradiology, that they're rarely used. And by clinical practice, I mean actually used for diagnosis, prognosis, or treatment management in individual patients. So our aim in this piece of our research was to deliver MRI biomarkers into hospital imaging departments, which could have two paradigms. Either we change the acquisition of the scans to measure these specific MR measurables, such as relaxation times, or we take standard of care images that are already being acquired and apply advanced analysis to them to extract imaging biomarkers from standard images. So, again, this is one of my public engagement slides. But the idea of quantitative neuroradiology is that the model now is that as a patient you attend for a scan. Your scans will be reviewed by a radiologist. The radiologist writes or dictates a report, which is received by your consultant, and then your consultant discusses the results with you. What we want to do is intercept the images before they get to the radiologist, take them offline and do some machine learning or advanced analysis on the results, generate what we call a quantitative radiology report, which is a graphical dashboard presentation of this quantitative information, and feed this back into the system so when the radiologist is reporting at their usual workstation and their usual workflow, they can see this information and use it to inform their radiology reports. That's what we're trying to achieve. To work on this, Tarek, myself, and at the time Sebastian Auslan, in 2015, set up something called the Quantitative Neuroradiology Initiative, and after Seb left, Frederick joined us to join the leadership team. And we really wanted to put some focus into how we could achieve this aim of quantitative neuroradiology. And we established a rational framework for doing this because we need to proceed in a systematic way and identify some of the issues we need to work on. My part in this really, one of the most important parts for me has been to lead, in the loosest sense of the word, some work to develop some software infrastructure to allow this to happen. One of the reasons it's challenging to get quantitative MR into the clinical workflow is are to do with networks and the information systems and how we actually get things into an automated system, because the days of being able to transfer data offline for someone's PhD student to analyse and then feedback for an MDT, it's really not how to proceed. We need automated systems that are integrated into the hospital workflows. So, and then this takes us into the domain as software as a medical device. And this was the piece that was missing back in my early days when I was doing that T2 measurement in the neonatal brain. The concept of software as a medical device wasn't really very prevalent then, and it is now, but this is how we proceed. So it's resource intensive. We had to get some funding from our BRC and from a Wellcome ISSF award. To pay for time of professional software developers, professional research software developers from the group that's now the Advanced Research Centre at UCL. And we set up a development team, which included these people here. And James in our group, James Mogridge, really led on putting this project together and making it happen in practical terms. And we developed the software to do this under a quality management system, which allows us to achieve regulatory compliance because there's an in-house exemption for software developed in this fashion. And we've deployed this software on our hospital network here at the National Hospital. And this allows us to deploy quantitative neuroradiology methods in our own service. But obviously, we're taking a very cautious approach because there are safety issues with this. If the analysis goes wrong, it could cause harm. So we still have a certain level of manual quality control. But this, as a piece of software development, is very useful. And it allows us to incorporate different analysis modules to support them in a containerized way by this QNI central engine, which is the interface between the hospital PACS system and the analysis. We've had one major success, which has had real clinical impacts locally with this, which is our epilepsy hippocampal sclerosis QNI report. This was based on work done originally, or led originally by John Duncan out at Chalfont in the epilepsy research team. And what it does is it identifies this area of the brain called the hippocampus, which is picked out in pink in those images automatically, and performs measurements on the size and the asymmetry of this structure, and also the T2 of this structure. And this information is used to inform decisions around the neurosurgical management of epilepsy. And we run this on scans from 2 or 3 patients per week. Within our service, and we've been doing this for quite a few years, so it is having real impact. However, there are limitations. It's limited to one MRI scanner. We now are stuck with having to maintain this software and this system ourselves. So it's not a perfect solution, but it certainly was a good exemplar project of what we can do as NHS clinical scientists and clinical academics. So what are we doing to take this forward? Well, we're working on new indications. Just a couple of examples. I'm very keen on trying to introduce this technology to benefit patients with neuromuscular diseases, to transfer those great successes in the research domain for fat water imaging for research studies, to bring that into the hospital as a measure to benefit patients attending for MRI scans. So we have some funding from the National Brain Appeal to take that forward. This is something that I'm working on with Jasper and other colleagues to take forward. We also, Tarek recently was awarded an MRC project grant to work up protocols for quantitative neuroradiology in movement disorders. And that's something we're working on with colleagues, including Karen Schmelly from Medical Physics, with whom I've had a collaboration around the clinical applications of quantitative susceptibility mapping for a number of years, and Ramona Wheel, who brings in the neurology expertise for movement disorders. So we've got some new things on the go. A number of years ago, under Tarek's leadership, we established our Quantitative Neuroradiology Innovation and Adoption Centre, which is based on the potential of our new Gray's Inn Road Imaging Neuroscience Centre, which is a collaboration between the NHN Academic Neuroradiology, UCLH Digital Health and UCL Engineering, recognising that all of these parties are required to achieve clinical adoption of these methods. And this is the current steering group. So this is an umbrella that we're using to help take forward a number of these projects. It's become part of the learning experiences that we can't do in the long term. We can't sustainably create, manufacture, and support these types of applications in clinical service ourselves. We simply don't have the resources. We don't have the software engineers, we don't have the support teams, we don't have the money. So this is something that we're looking to take forward with industrial partners. So we have our Siemens-Quinniac partnership, which is where we're exploring how Siemens can help us with this. For instance, they have platforms which have regulatory approval, which can support third-party analysis applications, and we're exploring that with them. But we have a very constructive partnership now directly with the Siemens, with the Siemens German factory team, and with the Siemens UK team who are named here. And in a couple of weeks, we're actually interviewing for a Siemens scientist who will be dedicated to supporting some of our work at Queen Square and then in Gray's Inn Road. We've also, over the years, been working to support standardization in issues, to look at how these quantitative measurements can be calibrated so they're consistent between centres, between sites, between scanners. And this has led to a fruitful and pleasant collaboration with our UK National Physical Laboratory, with their MRI experts, Matt Hall and Matt Cashmore, where we're looking at ways that the principles of metrology, of formal measurement science, can be used to try and improve the results we get in quantitative neuroradiology. So I've nearly finished. I just wanted to touch on, all of these things over these years have been very rewarding for me, and I've really enjoyed this time, and I've been privileged to be given opportunities from supervisors and collaborators to take all this forward. And you get to this stage, 35 years at UCL later, to think about how can I help create these sorts of opportunities that I've enjoyed for those coming up behind me. And I also believe that we as NHS clinical scientists can play a real pivotal role in this kind of direct translation of imaging technologies. I think because we have one foot directly where we're embedded in the clinical services of the hospital, but we also have another foot in the research community that we exist in. So we can literally be a conduit for translation. But we're very busy doing things that are not this kind of research and development. So we need to find ways that this work can be funded. And there are a number of ways this can happen. I'm very appreciative of the support I've had from my post from our local NIHR BRC, the Translational Neuroscience team, has funded fractions of my working time so that I can dedicate it to this kind of work legitimately. So this is one means that this can be achieved. Something else that I've been working on is to try and bring in research funding to help support our NHS clinical scientist posts. So we have two new posts that are part funded by research. We've recently very pleased that we've been able to appoint Marjana into a more senior role, which is a very deserved promotion. And this is with some part funding from John Duncan to support the research activities of the Society for Epilepsy. And the post that Jodie Smith will move into in October has some part funding from the neuromuscular side, from the LifeArc Centre for Rare Mitochondrial Diseases. So she'll be spending some time working on neuromuscular disease applications of these methods. So this is another way, and obviously we're looking in the future to see how we can do this more often. But I think the most powerful is the idea of establishing new permanent posts, clinical academic posts for MR physicists. And we have a model for this. A number of years ago, I worked with our divisional management to enable us to contribute 50% of an existing post that we had at Queen Square of a hospital post, to combine it with a 50% of an academic post that Danny Alexander had in computer science, to set up a new post, which served the was the first post holder, was very successful, and now Matt Grexellars is in the post, and very successfully, all credit to him, spending half of his time being a UCL associate professor, doing research, academic things, and pursuing translational research within the former CBIC, and working 50% time as a member of our clinical sciences team, providing essential contributions to our clinical departments from the clinical science side. So this has been very successful, and we really want to scale this and create some more posts of this nature. And I'm hoping that through the Grazing Road initiative, when we should be able to establish a new NHS physicist post, that we'll be able to establish similar partnerships with other academic partners, where we can show that we have really good alignment between the prevailing strategic priorities of academic partners and ourselves, which in this case was true because it was on the back of an AI funding boom at the time. But we're looking for ways we can align potentially with the priorities of the medical physics departments, the academic medical physics departments, or indeed the Institute of Neurology. So that's really the end of the talk. But I need to say some thank yous, as is traditional. I have to thank the radiographers, consultants and admin staff of the Elysium Department of Neuroradiology. I'd like to be able to mention everybody, all of you that are here, and all of the others, because there are so many, but I can't mention everybody. But obviously Tarek Yousry has been a collaborator and mentor now for 20 years, and we've, I think when I first arrived, people might have thought that we looked like people who would never work together, because I was a bit of a scruffy lab rat, and Tarek is Tarek. But actually, we do see the scientific world in quite the same way from a number of perspectives, and therefore have been able to form a successful collaboration, both building up the clinical sciences team and building up the technical facilities of our clinical radiology department and pursuing research. Susan Wakeling, who many of you know was our former services manager, was also a collaborator, friend and mentor before she left. And she really helped me with all of the personnel management and the human side of making these things happen. Caroline Andrews was the MRI superintendent when I first arrived in the department. And she helped me through the culture shock. She really taught me about adult neuro MR, which I knew nothing about at the time. And then there's our current radiographers, Lisa and her senior colleagues, and Helen helps with the research scans. And Ahmed, who's our current consultant MR lead, and Satiras, who was before, the clinical scientists, and everybody else. I can't thank everybody, but it's been a pleasure working in this department for all these years and continues to be so. On the UCL side, there's all my colleagues in academic neuroradiology. There's Karen in medical physics. There's the senior staff in the UCL Queen Square Institute of Neurology. Michael Hanna, who's always our director, who's always been encouraging. David, who supported me in my promotion application. Judith, who's helped with all the grants, the professional staff, and the education team. I've not had time to talk about the teaching that I was involved in, but as David said, I've been deeply involved in teaching for at least 15 years here at the Institute of Neurology, and the education team have been great. And then there's my colleagues in IPEM, which is our national professional organisation. And with these colleagues, not only have they been a very supportive professional network, but we've been able to promote some changes at a national level which have had impact on MR physics in medical physics services and actually benefited our community quite strongly. So that's a story for another time. And finally, my parents, Cynthia and Peter, who are watching online or will be watching the recording, who unfortunately couldn't be here today, but have always encouraged me through all these stages and their values of service and the importance of teaching have informed a lot of what I've done. And then, of course, last but not least, my wife Sharon, who's here today. This is our pandemic wedding a few years ago. But the truth is, Sharon has been providing essential enabling support for a lot of years before then and continues to do so. So thank you, Sharon. So that's really the end of my talk. And I'll leave you with some sage advice from magnets, bulbs and batteries, which has served me well over the last, well, since I was 8 years old. Thanks very much.
Thank you very much, John. To the audience, I think, like me, we enjoyed very much your presentation. We saw the evolution of a boy playing with transistors in a way that many boys play, but then moving on to make this his main area of interest and work, and moving on from that to a journey where patients are at the center of what you are doing, but never leaving the other part aside. I think we have seen a man really dedicated. We have seen a man who is quiet, softly spoken. He's not imposing himself just like that, but you realize very quickly that it is sensible to listen to him. What he says is relevant. It may not come across very powerful, but if you hear it, it is very, very powerful. So that was also my journey with John. I never looked at, I never realized that maybe from the outside we look a little bit of a funny pair, but maybe. But I realized very quickly that it's important to listen. And John is quiet. He doesn't say much. You wait, you wait, you wait. Did he listen? Did he not listen? You don't know. And suddenly he comes back with something that's quite important. And I learned not to move forward without having talked to John and listened to him. So that's one thing about John, that he's very collaborative, like actually many of the physicists, clinical scientists. They like to share, they like to work together, and we profited a lot from that. In his presentation, he was, as he always is, very precise, very clear. What he says and gets across is very powerful. He's one of the very few who have bridged the physics, the patients, and the research. So a clinical scientist and also an academic. We had here a few weeks ago the Vice Provost who mentioned that one of the most important things in someone becoming a professor, which is at UCL a challenge. It is much more of a challenge if you are mainly a UCLH employee and not a UCL employee. And he mentioned that the most important thing is, it's maybe my word, is tenacity and patience and the strength to go through the long way of work. There are always moments when it becomes more difficult, but you have to have this tenacity, and John had that. And he changed the course of many areas. So the first of consultant clinical scientists at NHNN, created the first academic clinical scientist post, which we didn't have before, modeled on, as he showed, the clinical academics, neurology, neurosurgery, and so on. So that's another first. And as a professor employed by UCLH, another first. So a lot of achievements, a lot of things to admire in the way you did it, and also the working together with the various groups, with the neuromuscular team, which really you pushed forward on the physics side, and also with all the other teams that you mentioned before. Safety was from the beginning something that was very important to you personally, but also important to the department that you also pushed forward. So pushing forwards in multiple areas, and finally now with the Q&A and with the Quinniac. It's a marvelous journey, which you showed us, and a marvelous journey for me personally. I always enjoyed working with you. I always take your opinion into account and take it very, very seriously. And it's been a pleasure, and I hope you all enjoyed this, and I would like you to join me in thanking and congratulating John for his journey.
So just a very brief final word from me. Thank you, John, for a wonderful lecture. I really, really enjoyed it. Incredible range of diseases that you've worked on, and really putting all that physics experience into practice. Quite emotional when I saw that you grew up in Dorset and had the same books as me, the magnet bulbs and batteries and the newts and all that. I was tears in my eyes as I heard all that, but unfortunately none of my experiments worked at all, so I've had to stay well clear of anything to do with physics. But no, a wonderful inaugural lecture, wonderful journey. And really on the cusp of true clinical translation and implementation on a big scale. So we need more of you, don't we? We need more of these posts, more academic integration. And thank you, Tarek, for the wonderful vote of thanks. Perhaps an unlikely partnership, but clearly very productive and successful. So thank you all for coming. I believe there are some refreshments outside, and we can chat further over a drink. Wonderful to see you all. Thank you.